me hacking through this lecture. It's going to be better than like having a TA come or telling everyone just to go home and listen to the audio, so this is better, but you'll just have to bear with me through some coughing. So, quick announcement, a reminder, the midterm exam covers up to and including the end of today's class, so that's the end of today, right, end of the 3 hours. Previous midterm has been posted. We recommend that you do not just study from the previous midterm, because you're not getting the same questions twice, right? You should study the lecture material and the textbook readings. Both are testable. Use the previous midterm to test yourself, and then study some more. So do use the old midterm as an opportunity to test yourself, to use it as a measuring stick to make sure you understand the material correctly, and that you can do the midterms in the right amount of time, right? A lot of students like to look at the old midterms and just kind of look at the answers and stuff. No, use it as a chance to test yourself. Okay, so the upcoming schedule is as follows. So it's pretty easy up till about the fall break time. So October 20th, that's today, you're in the lecture, nothing to worry about, just stay here. October 26th is the midterm exam, and then 27th lecture, November 3rd lecture. November 10th is fall break, so no lecture that week. No, November 17th lecture. So fall break, it's great, right? I get to stay home, you get to stay home. But during the morning session, so I see you 0 times that week. During the morning session, they will see their professor for 1 hour, so we'll essentially be 1 hour behind the morning class. Now, there are 2 ways by which we can make this up. You don't need to decide now, we'll talk about it next class. 2 ways by which we can make this up, again, we're 1 hour behind, is that one option is to do 30 minutes more on November 3rd and 30 minutes more on the 17th. The problem with that is that you're going to be here for 2.5 hours of lecture here and 2.5 hours of lecture here, so it's pretty long. The second option is that we wait until the very last week of class. At the very last week of class, it's a bit of a shorter lecture. So in the very last week of class, you would have 2.5 hours of lecture. So the problem with that is then that you don't catch up till the very last week of class, right? And then there's less time to ask, say, the professor questions, since that's the last time you see me before the final exam. So both options are possible. So today we're going to do the lecture. We'll do a quick vote next class. Think about what works best for you. If you ask me, what I find works best for students is that if they do an extra half hour here and half hour here, rather than to wait for the very end. But again, check your schedule, see what works best for you, and then we'll just do a vote on October 27th next week to see what works, okay? So keep that in mind. So this week is glycosylation. The first lecture is about intracellular vesicular transport, which will translate into the second lecture. We'll talk about that some more. Do your readings as per usual. First thing we want to talk about is glycosylation. So imagine adding sugar groups, right? Glycosylation, like sugar chains to a protein. Most soluble and transmembrane proteins in the ER are glycosylated. So soluble as in the ER lumen, and the transmembrane proteins, you know how those transmembrane proteins now are inserted into the ER. So the soluble and transmembrane proteins in the ER are glycosylated. Cytosolic glycosylation can occur, but it's very rare. So cytosolic glycosylation, it can occur, but it is very rare. And it's very simple. So we're not going to focus on that in this particular class. We'll focus on the ER one. There are 2 types of glycosylation when it comes to this ER glycosylation. There's O-linked and N-linked. O-linked means you're adding the glycosylation onto OH groups, hydroxyl groups. So say you're adding the glycosylation onto serines or threonines of proteins, onto OH groups, like serines with threonines. Now you also have N-linked glycosylation, where you add the glycosylation onto asparagines. So N for NH2, onto asparagines. N-linked for asparagines onto NH2 groups. O-linked is very rare. Again, O-linked is about 10%. And that means N-linked is about 90%. So in this class, we're going to focus in on N-linked But remember, there's O-linked as well. For the N-linked oligosaccharide precursor, it's pre-formed in the endoplasmic reticulum, and then it's going to be linked to the target protein in the endoplasmic reticulum. Okay, so to review, most soluble and transmembrane proteins in the ER are glycosylate. You can have glycosylation in the cytosol, but ER is much more common. For the ER, there's two types. O-linked, only 10%, where you're adding the glycosylation onto an OH group, like of serine or threonine. For N-linked glycosylation, it's for asparagine, NH group. The N-linked oligosaccharide precursor is pre-formed in the ER, and you link it to the target protein in the ER. So here is your sample protein, N-terminus over here, C-terminus over here, many amino acids. One of them is asparagine. Notice this is asparagine. There's an X, and then there's serine or threonine. You'll see that in a sec. But on the asparagine, on its R group, on the NH, you can add this oligosaccharide that is pre-formed. Now, you don't have to memorize the exact configuration of this oligosaccharide. You can see it's quite complicated. It's actually the same one that's added onto all these asparagines, and you'll see how it's added in a sec. Look at the composition here. Again, don't need to memorize the exact composition, but take a look. In red, you have N-acetylglucosamines. In blues, you have mannoses, and in green, you have glucoses. So this is the same thing that's added to many asparagines. What you'll see in a sec, you need to write this down right now, but what you'll see in a sec is that this one large glycosylation group, this huge sugar chain, is going to be modified, and sometimes it's modified so much that all that's remaining is this gray part. So lots and lots of modification can occur. Sometimes the only thing that's remaining is this gray part, the gray core over here. In the endoplasmic reticulum lumen, the N-linked oligosaccharide precursor is transferred by an enzyme we call oligosaccharyl transferase to an asparagine on a protein being synthesized. Now, the oligosaccharyl transferase will look for a sequence ASN serine or ASN or X threonine, where X is any amino acid except proline. So you actually look for this pattern. Asparagine and X serine, asparagine, X threonine. If you see this pattern where X is any amino acid except proline, you're going to add that oligosaccharide chain onto it. And proteins are only glycosylated on the ER lumen side, right? We're talking about the ER lumen, not the cytosolic ones that are very rare. Okay, so if you take a look at your diagram, this is quite similar to the diagrams we took a look at last class. So this is the endoplasmic reticulum. Here's the ER lumen. Here's the cytosol. Here is your protein. Again, this is your transporter. You can see in red, this will be an internal start transfer sequence. And you can see the NH2 is facing the cytosolic side, so this is probably more positive. It's probably more negative here. And as the ribosome is making it, you are feeding the protein into the ER lumen in this case. And not shown for you here is going to be the ribosome and the mRNA. Now, as you're feeding it in, you have this membrane protein here, oligosaccharyl transferase. And notice on the ER lumen side, it's scanning this. It's looking for ASN X serine, ASN X threonine. When it finds one of those, it will transfer this whole chain over to the ASN looking like this. So notice this chain is pre-formed, and the enzyme simply looks for the right pattern of amino acids and will transfer it over to the asparagine. Pretty simple. After you have transferred that oligosaccharide onto the protein, 3 glucoses will be removed. That's linked to proper folding of the protein, which you'll see. Then 1 mannose is removed. Upon that, after 3 glucoses and 1 mannose is removed, then you can transport that protein to the Golgi apparatus. So here's that diagram. Asparagine, again, X serine, asparagine, X threonine, right? That's your protein. You have added that huge oligosaccharide chain onto it. Now, as it says here, you're going to remove 3 glucoses and 1 mannose. This is how it's done. Glucosidase 1, it's a specific enzyme, will remove 1 of the glucoses. Then glucosidase 2 will remove the second glucose, and then come back and remove the third glucose. Be a little careful here, because if you look at this, it looks like glucosidase 2 chops off 2 of the glucoses. Not quite. Glucosidase 2 will chop off 1 of the glucoses, and then it goes away, and then it comes back again and will chop off another glucose. So it's an iterative process. It's not like it just chops off 2 at a time. Does 1, then we'll do the other one. Then an ER mannosidase will chop off 1 of the mannoses. So if you look at your standard chain, again, you don't have to memorize how to draw this, but you can take a look here. You're going to remove 1 glucose, then glucosidase 2 will remove the second glucose, then glucosidase 2 remove the third glucose, then ER mannosidase here removes the blue one. If you match these up, the blue one that's missing is going to be off this chain right here. It's going to be this blue one right here that is missing right here. Okay? So after you have those removed, now that's a signal that says this protein is ready for transport via vesicles to the Golgi apparatus. When you go to the Golgi apparatus, the different cisterna, the different sacs, are going to have different enzymes to process those sugars quite differently. Okay, so here is your standard Golgi apparatus. We're going to go through the setup of the Golgi in a bit, just to remind you, just to orient you. We'll talk about this in a bit. But facing the endoplasmic reticulum side, we have cis Golgi, that's the side that faces the ER. The opposite side is the trans side, and in the middle is the medial. So in these different Golgi sacs, in these different membranes, you have all sorts of different enzymes. And depending on how you need to process that oligosaccharide, you're going to send it to different sacs, right? So all the different sacs will remove or add sugars, depends on what the protein needs. And you can see an example here. Here is the N-linked oligosaccharide precursor that gets added on in the ER. 3 glucoses and 1 mannose will be removed, and then you'll process it in the Golgi. You can see it gets processed quite insanely, right? Like you can see, you remove 3 glucoses, you remove 1 mannose. This is what you get. Then when you go to the Golgi, lots and lots of processing can occur. Depends on the protein, depends on the oligosaccharide chain. Okay, so just to summarize, so you don't need to flip back, but I just want to summarize what we've been through so far, because it gets a little bit complicated. Again, many kinds of glycosylation. We're focusing in on the N-linked. And we said for N-linked, you add this preformed chain onto an asparagine. We said as you're sending the protein, whether it's a transmembrane protein, whether it's a luminal protein, this oligosaccharyl transferase is looking for the sequence ASNX serine or ASNX threonine. When you see it, you add that huge chain onto the ASN. And then after you add it, well, in the endoplasmic reticulum, not in the Golgi yet, still in the ER, you will remove 1 glucose, then you remove a second glucose, then you remove a third glucose, then you remove a mannose. Now it's ready for transport. Then you leave the ER.